in this live session, we are going to talk about the most important concepts of genetics. And here we are going to discuss about the six important concepts. First one is multiple allelism, second one is pleiotropism, and the third one is polygenic inheritance. Now, next is the codominance, incomplete dominance, and epistasis. So these six concepts are the most complex concepts in which each and every student gets confused. But no worries today, because today we are going to clear all these six concepts in this very session. Okay, now guys, before explaining and going to these six concepts or which are the Mendelian deviations, we will just start from a beginning. So let me tell you what is a gene. So guys, gene is nothing but a single DNA segment, or a small DNA segment is known as a gene, which carries your hereditary information. Which carries your hereditary information is termed as a gene. Okay. Now we can draw a gene. How can we draw the gene? We can draw the gene like this. A gene has two parts. Okay, and these two parts are known as alleles. Alleles, or we may also say these are the allelomorphs. Okay, these are the allelomorphs. Now, for this one is the first allele. Okay, so this one is the first allele, whereas this one is the second allele. So a gene is made up of two parts, which are known as two different alternative parts. Now these are known as alleles, or these are known as allelomorphs, and these are, suppose we can name this as capital A and another as small a. Then this first one will be said as the dominant allele, whereas another will be said as the recessive allele, right? Okay. Now, when in an heterozygote, what happens that the these type of Mendelian deviation are not these type of Mendelian deviation are not followed. That is, the dominant and the recessive relationship are not followed. Then these type of Mendelian deviation codes for multiple allelism, pleiotropism, polygenic inheritance, codominance, incomplete dominance, and epistasis. Now, guys, let me tell you that when gene has two parts of it, that is the alleles. Now alleles will have a different function. Okay, dominant allele will be expressed when present in an heterozygote, whereas the recessive one will not be expressed. Clear? The dominant allele will have the activity to suppress or to ignore the activity of the recessive allele. Okay, now guys, before going to the most important concepts, let me tell you these concepts are done through the gene interaction. So one gene when interacts with the other gene, or one allele when interacts with the another allele, then there forms the gene interaction. And these all the phenomenon, that is multiple allelism, pleiotropism, polygenic inheritance, codominance, incomplete dominance, and epistasis, are all the phenomenon which falls under the gene interactions. So these are all because of the gene interaction. Now guys, gene interaction are of two main types. One is intergenic, okay, and another is intragenic. Okay, now what is intergenic, guys? Let me tell you about a very easy example, that is inter-school competition. So what do you mean by inter-school competition? When there will be competition between your school, okay, when there will be a competition between your school and your friend's school, okay, when there will be a competition between your friend's school, suppose B, okay. So when there will be competition between school A and school B, then this type of competition is known as inter-school competition. It is known as inter, I-E-I-N-T-E-R, inter. Similarly, that is when there will be competition between the genes, okay, between two separate genes. When there will be competition, suppose in between the capital A and capital P, okay, when there will be competition between two separate genes, then it will be known as intergenic in gene interactions, okay. But guys, when there will be competition between the same gene or between the two alleles, when there will be competition between two alleles, then it will be known as intragenic. That is within. Intra means within and inter means between, okay. So what do you mean by the word inter? Inter means between, okay, and intra means within. So when there will be competition between two alleles of a gene, when there will be competition between capital A and small a, then it will be known as intragenic gene interactions. Clear? Now, in case of the intragenic gene interaction, we are talking about one character. Okay. Suppose, let me keep like this, intragenic. This one is intragenic. This one is intergenic. Okay. Now, in case of intragenic, we are talking about one character. Will talk about one character. Now this gene, that is gene A and capital A and small a, will only code for one character. Suppose this capital A will code for one character, and this PP will code for the another character. Okay, another character. Now, what will happen, guys? That okay, let me show you first. So what will happen that in case of the intragenic gene interaction, we are talking about a single character and we will also get a single result. Okay, we will get just a single result. So this, when we are talking about one character, then we will just go on for the codominance, incomplete dominance, and epistasis. So these three are the intragenic gene interaction. Okay, when there is only one character. In codominance, we talk about the color or we talk about the blood group, right? Only one character. In incomplete dominance, we only talk about one character, that is color of the flower, okay, color of the flower. So only one character. In epistasis, we talk about the coat color, okay, that is color of the body fur. So there is one character, or we can see only one character. But in case of intergenic, what we see, guys? We see more than one character. We talk about more than one character. Okay, when we talk about more than one character, that means two genes will be interacting and we will be getting something different. Suppose when your the gene for the green or the gene for the yellow pods is being taken, and along with the round seed is being taken. Okay, so yellow and the round seeds, when we talk about both the characters here, two characters are present. We are talking about two characters, right? So when we are talking about two characters, then there is the interaction between two genes. So what are the two genes? One gene is coding for the color, or one gene is coding for the yellow color, and another gene is coding for the shape of the seed. Okay, one gene is coding for the color of the seed, whereas another gene is coding for the shape of the seed. Clear, guys? So here we are talking about two characters. That means two different type of genes will be present. One will be the Y gene, one will be the R gene. Okay, so these type of, that is intergenic gene interactions, are the multiple allelism. Okay, these are the multiple allelism. And then wait a second, guys. Okay, so these so okay, so these are the multiple allelism, paleotropism, and polygenic inheritance. So what we got to learn? We got to learn that multiple allelism, paleotropism, and polygenic inheritance are the intergenic gene interactions, whereas codominance, incomplete dominance, and epistasis are the intragenic gene interactions. Is it clear, guys? What is intragenic? What is intergenic? Now we will move to the first topic, that is multiple allelism or multiple alleles. Now, what are multiple alleles? We have already studied that one single character or one single trait. So, one single trait is being controlled by one single gene. Okay, what did Mendel say? That one trait is controlled by one gene. That means genes are very specific in controlling or in coding for one particular thing. Okay, and but in case of multiple allelism, it is a total deviation from Mendelism. What we get to learn that alternative forms of a gene are very often available, and the alternative forms of a gene are called as the alleles, which I already told you that what are alleles. Let me again draw it for you. Suppose this is a gene coding for capital A and small a. Then this one is an allele and this one is another allele. Number one, number two. I hope now you can understand what are alleles. And this total is a single gene. Okay, this total is a single gene. Now, when the number of alleles become more than 2, when the number of the alleles will become more than 2, suppose there will be in normal condition there are 2 alleles, but in abnormal condition there can be more than one allele. Okay, there can be more than one allele. In case of a normal male chromosome, okay, but in case of the Down syndrome, what is there? XXY. So one more chromosome is present, one more gene is present, one more allele is present. So when the number of the allele will become more than 2, generally 2 alleles are coding for a single gene. When there will be more than 2 alleles, then they will be known as the multiple alleles. Then they will be known as the multiple alleles. If it is clear, then please do note down in the chat section. Please do tell me in the chat section, yes ma'am, it is clear. And make sure you like this video, share this video with all your friends, and clear all the doubts, right? And genetics, and after this, watching this session of mine, you must say that yes ma'am, I just love genetics. Okay, now coming to the example of multiple allelism, which is very important, another most important, and also it is very important for your board exam. It is also very important for your NEET exam. It is almost said to be a NEET bomber. So the example of multiple allelism is ABO blood group in the man. Now ABO blood group system in the man have 4 main type of phenotypes. Okay, we can see 4 main type of characters. One is the O type blood group, another is the A type blood group, next third one is the B type blood group, whereas the fourth one is the AB type of blood group. And this O type of blood group is the recessive one. Okay, it is the recessive one, and AB is the dominant one. Okay, AB is the dominant, whereas O is the recessive one. And so you can see it is being coded by a recessive gene, that is small I small I. In case of blood group, there is a gene which codes for the blood group, and that is the I gene. Okay, but that is the I gene which codes for the blood group. And this one I gene is the dominant one, and so what will be the recessive part of the I gene? That will be small I gene. Okay, so as O is the recessive one, so O is coding for the small I small I. It is homozygous recessive. Okay, and what are the heterozygous alleles in present in the case of the blood group? Heterozygous can be found in the type A and also in the type B. Okay, so type A can be homozygous, that is it can be IA. Okay, or it can be, in case of heterozygous, it will be IA and small I, where IA is dominant. Here IA is the dominant allele. IA is the dominant allele. And in case of B, similarly, IB is the homozygous, whereas IB small I is the heterozygous, and here IB is the dominant allele. Again, a case we can see here in the ABO blood group system, which is very important, guys. See here, IA is also present, IB is also present, right? Now it could have been only one could have been dominant, that is either A would be dominant or either B would be dominant. But this doesn't happen. So what happens actually? Both of them are fully expressed in a heterozygote. So it is also heterozygous condition. It is also a heterozygous condition, and in this heterozygous condition, we can see both the alleles are dominating and both are fully expressed. Both are fully expressing itself. Neither A is leaving B, neither A is suppressing the activity of the B, or neither B is able to suppress the activity of the A, and both are fully expressed in this type of heterozygous condition, and this is known as codominance, because both are showing their power, and then they both are dominant in nature. So these are known as, both are known as codominant. Okay, now this is a very important example, guys, and just make all the stars you can make in this type of example, because it is a NEET bomber and it has been asked, and it is always asked in the NEET question. Even in the previous year, you can see the assertion reason question from these multiple allelism, right? Assertion reason question can be like this, that is every multiple allelism is an example of codominance? क्या सारे multiple allelism का codominance के example हैं? अगर yes, so you have to go for the option number A, that is assertion is true and the reason will be the correct explanation. But the assertion is totally false, guys, because every multiple allelism, suppose this small I or the A type or the B type, all the are not the example of codominance. Only AB type, only the specific AB type is the example of codominance. Only the specific AB type is the example of codominance. So nowadays, in the correct incorrect of questions, these type of questions are asked in the NEET examination. So make sure you know about this. Right now, let's move ahead. We have completed multiple allelism, so I hope it is important that you have noted down each and everything in your copy. And if you have not noted down till now, then please do note down, and please do subscribe to our amazing channel, that is Anamika's EduSpace, and we will be back to back. There will be live session in this channel, and you can get all the contents which are easy available and easy understandable to you and all your friends. Okay, so please do share this video, like this video, and comment down. If you have any of the doubt, then please do comment down and let me know all your doubts in the doubt section. So let's get ahead and let's go to the next point, that is incomplete dominance. So guys, we have already cleared about multiple allelism. Now we'll go and clear what is incomplete dominance. So incomplete dominance, from the word only we can understand that it is not fully dominant. Okay, it is not fully dominant. So it is said to be incompletely dominant. So that dominant gene is not fully dominant, or not fully or not fully able to replace the activity of the recessive gene. Okay, so here also there is a heterozygous condition. This condition also is a heterozygous condition, because there must be presence of two different types of alleles, right? Two different types of alleles must be present there. Types of alleles. Write with my as I'm writing here, you also write with me, so that you can also cope up with me, and after the class you can just download a PDF, or you can just have the your screenshots out from this slide. Okay, now the individuals with contrasting feature when produce some intermediate phenotypes. I have already said that the phenotype will not be fully dominant. So what did we studied in Mendel? That if there was a heterozygous condition like this, that is one capital T and one small t is present. Capital T is coding for the tall character, whereas small t is coding for the dwarf character. And in case of Mendelism, what did Mendel say? That when the dominant gene or dominant allele is present, then there will be no problem. The dominant allele will express its dominance. Okay, express its dominance, and it will repress the activity of the recessive gene, and it will show the phenotype of tall character, and it will show the phenotype of tallness. But it is also a deviation from Mendelism. It is also a drawback which Mendel did not notice, and this drawback is said to be incomplete dominance. Right, guys? Now, where do we find to see the incomplete dominance? So, we find this incomplete dominance to be seen in Mirabilis jalapa. Now, what is Mirabilis jalapa, guys? It is a flower. So, it is a flower. It is a night flower, and in the step of flower, there is a gene which codes for the red flower, as you can already see, that is homozygous red. This is what this one is, your homozygous. Okay, both are capital R, capital R. Capital R is denoting for the red color, whereas small r is denoting for the white color. Okay, smaller is denoting for the white color. This one is red and this one is white. And what happens when the parental strain is taken? Suppose, let me take a parental strain and let me show you one cross quickly, right? Okay, so we will take a parental generation where they will be present of homozygous parent. Capital R will be crossed with small r, small y. These are pure white, these are pure red. Now there comes the gamete generation. Gamete will be originated only one, because there is no heterozygous condition, there is homozygous condition. So only from these parental gene, only one gamete will be coming, that is capital R. From these parental gene, that is small r, only small parent will be coming, that is coding for the small r. Now in F1 generation, we will find one heterozygote. Right, we will find one heterozygote that is coding for capital R and small r, because two only gametes are present. One is capital R and one is smaller. Now, in case of Mendelism, if it would be a Mendelian cross, if it would be a Mendelian cross, then we would say that the color would be red, or the flower color would be red, may it be the heterozygous condition. But it is not a Mendelian cross, and it is a deviation from Mendelism. And what we got to see an intermediate color, that is pink. We got to see an intermediate color, that is pink. So pink color is the intermediate color between the red color and white color. So when you take a color palette, you can mix those color. You can mix the white color and the red color, and you can find that pink is the intermediate color between the white color and the red color. Okay, guys. So pink is the intermediate color. So that's what Mirabilis jalapa gives us an example, that in the F1, now when this F1 phenotype was set, when the F1 phenotype was set, that is F1 cross F1 was done, we get to see a phenotype, that is one becomes fully dominant, that is homozygous condition, one becomes fully recessive, and another, that is a heterozygous condition. Two type of heterozygous phenotype are seen, which obtains the pink color. So the ratio, or the phenotypic ratio, which is very important and asked several times, make as many stars as you can make in this type. So if phenotypic ratio of the incomplete dominance becomes 1:2:1. Okay. So phenotype of the incomplete dominance becomes 1:2:1. That is say that neither the red color is fully dominant over the white color, nor the white color is said to be fully recessive. Okay. So what we find, that capital R codes for the red color, but in case of a heterozygote, when both the conditions are present, that is capital R and small r is present, the red color is not fully dominant. Not fully dominant. We find the red color is not fully dominant. Clear? So this was the phenomenon of incomplete dominance. Now we will move to the next thing. So before moving, if you have any doubts, please note down in the comment section, and let's move. Okay. So now we will study about codominance. So guys, after a little water break, I'm back again. So codominance is another genetical phenomenon. Okay, when both the alleles of a trait in a heterozygote are fully expressed, as we have seen in the ABO blood group system. So IA and IB was fully expressing. Neither A was dominant, neither B was dominant. Both were expressing its dominant power. Both were fully expressing itself. So when both the characters are fully expressing itself, then together both the substances appear in a heterozygote, and this type of condition is known as codominance. Okay, now what happens in this type? This is also a Mendelian deviation. Okay, so let me. Okay, so this is also a Mendelian deviation. Right? Now, what happens in this phenomenon? Both the dominant and the recessive alleles lack their dominant and recessive relationship, and both have the capability to express themselves phenotypically in a heterozygous condition. So, the dominant and the recessive allele doesn't have any dominant and recessive relationship among themselves, and both of them expresses themselves equally. Okay, both are expressed equally, fully expressed, equally expressed. Now, in case of the coat color, okay, in case of the coat color, we see this type of codominant character. So one coat color was for the CW, and CW is coded for the white fur. Okay, white coat fur, and this one was white fur. Okay, fur color was white. This one is a red fur, red color fur. In case of the cows, in case of the cow, we can find white cow, we can find red cow, and then what happens when a fully white fur cow or a fully dominant white cow was crossed with a homozygous red cow, then what we obtain? We obtain a intermediate color, not intermediate actually. We obtain a different color which is neither intermediate nor it is a dominant color. That is neither it is white nor it is red nor it is an intermediate color. We just obtain a different color which is a roan color. Roan color almost is like a brownish color. So if you mix the red and white, if we mix the red and we mix a white color, then we will obtain an intermediate color, that is pink, which we see in the mirabilis jalapa flower. But in case of this type, we see that in case of codominance, the pink color was also not obtained. So what type of color was obtained? A roan color was obtained, which is a brownish, dark brownish color, and this brownish color is known as a roan coat color, which is again codominant, because here the W, that is the CW gene, and the CR gene, that is both the alleles are codominant. Both the alleles are fully expressing itself. I hope it is clear. Let me know in the comment section. Comment section में एकदम आंसर्स आने चाहिए फटाफट से. आगे बढ़ते हैं नेक्स्ट क्वेश्चन पे, सॉरी नेक्स्ट टॉपिक पे, that is pleiotropism. So what do you mean by pleiotropy? So pleiotropy is the phenomenon or the genetical phenomenon where there is a single gene but there is more than one phenotypic effect. Okay, so what did Mendel said? Mendel said one gene, one hypothesis was coded, and one gene, one hypothesis was also obeyed by Mendel. It was one gene was one hypothesis was also obeyed by Mendel. What Mendel said, one gene will only encode for one character. But in the pleiotropism, we find that one gene is not coding for one character. One single gene is coding for more than one phenotypic character. Okay, one single gene is coding for more than one phenotypic character. Suppose we take the for an example, we can say in Drosophila melanogaster, which is a fruit fly. Okay, almost looks like the house fly you have in your house. It almost looks like the house fly. So it is a fruit fly, Drosophila melanogaster. It is a scientific name. So it must be, when you are writing it, it must be underlined separately, and the binomial nomenclature must be maintained. So Drosophila melanogaster is a fruit fly, and this fruit fly is having a gene which is encoding for their white eyes, for the presence of the white eyes, and it is also affecting the sperm storage organ in the females, as well as it is also affecting some other structures. So these type of genes are known as pleiotropic genes. Okay, so what are pleiotropic genes? Pleiotropic means one gene and many effects. There will be one gene, only one gene will be there, but there will be several effects. One gene can control the color for the white eyes. It also can control the storage of the sperm in the sperm, or the sperm storage organ in the females, right? So it is controlling two main types of effects, and it is also controlling other effects too. So this is known as pleiotropism. Okay, now in pleiotropism, what happens, these genes, which in addition to their main effect, the main effect for this gene was, for the fruit fly gene, was to control the white eyes, but in addition also it is also acting as a modifier for another but entirely different gene. Okay, so the gene for the white eyes is never related, or more or less far too near, it is never related to the sperm storage organ. Okay, sperm storage organ is a totally different and entirely different character, which is controlled by the white eyes gene only, which is controlled by the similar same gene only. So I hope it is clear. जल्दी से chat section में smiley बनाओ और फटाफट आगे बढ़ते हैं next topic पे, जो कि polygenic inheritance. तो प्लियोट्रॉपिज्म की बिल्कुल ऑपोजिट फेनोमेनन है पॉलीजेनिक इनहेरिटेंस. वहां पे क्या था, वहां पे था one gene many effects, and in this case we can find that many genes will be there, many genes, more than one genes will be there, but effect क्या होगा, सिर्फ एक ही effect है, so only one effect will be there. This type of polygenic inheritance can be seen in the skin color, or the presence of the melanin content in our human body. Okay, the melanin content which is present in the human body. Skin color, it codes for the skin color. Very best example you may say, which is present in the human body. Okay, now in this case we can find several, total 7 phenotypes we can find. Okay, total 7 phenotypes we can find. Let me show you a graph, and in this graph you will be able to know this. Let us take another color and let us make it more and more interesting. So these ones are the darker one. So dark, very dark, and then are the darkest. Okay, here we'll get light, very light, and lightest. Okay, this will be the mulattoes, and this will be the albinos. So white are known as the albinos. Okay, these are albinos are termed as completely white. Albinos are completely white, whereas darkest or the mulattoes will be the darkest black. Okay, now what happens when a trait is determined by more than one gene? So how many type of phenotype we can see here in the case of skin color in human? We can see 7 phenotype. Okay, we can see how many types of skin color can be there. The skin color can be darkest, the skin color can be very dark, skin color can be dark. So 1, 2, 3, and it can be light, it can be very light, and it can be lightest. So 1, 2, 3. So 3 plus 3, 6, and 1 intermediate color. So mulattoes are the intermediates. Okay, mulattoes are the intermediates, and when there we will find the intermediate color, then it will be known as the mulattoes, and the completely white will be known as the albinos. Okay, and the totally dark are known as the negroes. Totally dark are known as the negroes. So guys, we can find total 7 phenotypes. We can find total 7 type of skin color in case of the human body. Okay, now these are controlled by some genes which are known as the polygenes. These are controlled by some genes which are known as the polygenes. So when a trait is determined by many or more than one gene, then its inheritance is known as polygenic inheritance, and the genes are called as the polygenes. For example, skin color in human. Okay, now let me say you that homozygous dominant, the total genotype working for this is ABC. 3 main type of genes are working for this. Okay, but the dominant or the darkest black, we can find the dominant capital A, that is more and more melanin pigment is present. Melanin pigment is more and more present in the darkest or the dominant person, or this person the melanin is higher, whereas melanin pigment is the lowest in this albinos, that is the completely whites. These are the intermediates which are the heterozygous. These are the heterozygous intermediates, and these are the homozygous dominant and homozygous recessive ones. I hope it is clear. You can find a clear picture of several skin color showing in your book. Okay, whichever book you follow. Next, coming to the last one and very, very important one, that is epistasis. Now, epistasis is a complete Mendelian deviation of the law, that is law of dominance and recessiveness. It is a complete deviation of the law dominance and recessiveness. Yes. So, it is a complete deviation. It is a total Mendelian deviation of the law of dominance and recessiveness. So what is epistasis means? Actually, epistasis is a Greek word. Okay, and the word and it means act of stopping. Okay, इसका मतलब क्या है? Act of stopping मतलब कुछ चीज हो रहा है, कुछ काम हो रहा है, कुछ एक्टिविटी हो रही है, उस एक्टिविटी को क्या करता है? ये स्टॉप कर देता है, रिप्रेस कर देता है. It represses the activity. So when there is an interaction between the two genes, and one gene is known as the epistatic gene. When there is the interaction between two genes, one will be the known as suppose, if you suppose see, one is known as the epistatic gene and another gene is known as the hypostatic gene. I'll tell you. Wait, what is hypostatic gene? What is epistatic gene? I'll surely tell you. Okay, so epistatic gene is the gene which is having the dominant power. Okay, now let me tell you, epistasis can be of two types. Okay, epistasis can be of two types. It can be dominant epistasis, which we can find in dogs, and it can be recessive epistasis, because the recessive gene can also function as a epistatic gene. Okay, when there will be, suppose when there will be the presence of a recessive gene, then this if there will be a presence of a hypostatic or dominant gene also, then also in the presence of the recessive gene, the dominant or the hypostatic gene cannot act. Okay, and in case of dominant epistasis, when there will be present of a capital I gene, then if there is a presence of dominant gene or either the recessive gene or the either any of the other gene, then the hypostatic gene will never be able to act. Okay, so two types of epistasis is there. One is recessive epistasis, one is dominant epistasis. Okay, so what is the fact of epistatic gene? So that what happens, this is a interaction between two non-allelic genes, mean between the interaction between two genes. Okay, so what is it is termed as? It is termed as intergenic interaction. Okay, and it is present in separate loci, and one gene marks the marks. Okay, it inhibits or suppresses the expression of the another gene. Now, the gene that suppresses the other gene is known as inhibiting or the epistatic factor, and the one which is prevented from exhibiting itself is known as hypostatic factor. So, जो जीन एक्सप्रेस कर सकता है अपने आप को, उसको हम लोग बोलते हैं एपिस्टैटिक जीन. ओके, एंड जो जीन अपने आप को एक्सप्रेस नहीं कर सकते, उसको हम बोलते हैं हाइपोस्टैटिक जीन. So though similar with dominance and recessiveness, almost लग रहा है ना कि हाँ similar है dominance and recessiveness से, but this is totally different and the two factors occupy two different loci. Okay, so we can see the epistatis or the dominant epistatis ratio is 12 is to 3 is to 1. Okay, the ratio of dominant epistasis is 12 is to 3 is to 1, and the example is dominant epistasis is seen in case of the dogs. Okay, how? Let me explain with the example. So what happens? This is the body color of the dog. Okay, body color of the dog is taken. Let us go for the body color. This body color capital B is encoding for black color. Okay, when there will be presence of capital B, there will be black dogs. Okay, or may or the capital B small b present, then it is a heterozygous condition, then also the dogs will be black in color. When there will be small b, then the dogs will be white in color. Simple, very simple cross, very simple. Okay. When there will be recessive variety, then the dog will be white. When there will be dominant variety, the dog will be black, right? But what was seen that in the presence of another gene, that is the epistatic allele, as it is a dominant epistasis, so the gene will be also a dominant one, right? If it would be a recessive epistasis, then the gene would be of recessive one, that is the small I one. Okay. So in this, if there was a presence of a small I with the black color, then the black color will not be able to express itself. Okay. चाहे कितना भी ब्लैक कलर डोमिनेंट क्यों ना हो, अगर रेसेसिव एपिस्टेसिस के बारे में पढ़ाई करेंगे और स्मॉल आई वाला जीन अगर कहीं भी जीनोटाइप में प्रेजेंट हो गया, यहाँ पे सपोज आपके कैपिटल आई भी प्रेजेंट है या उसके साथ स्मॉल आई वाला जीन है, तो स्मॉल आई वाला जीन if we study about the if we study about the recessive epistasis, तो अगर स्मॉल आई वाला जीन कहीं भी प्रेजेंट आ गया ना किसी भी जीन जीनोटाइप में, तो क्या हो जाएगा आपका ये पूरी तरीके से मार्क्स कर देगा, चाहे कितने भी ब्लैक कलर प्रेजेंट हो, कैपिटल आई प्रेजेंट हो, कुछ भी प्रेजेंट हो, ये अपना कलर एक्सप्रेस नहीं कर पाएगा। ठीक है, ये रेसेसिव एपिस्टेसिस के टाइम पे स्मॉल आई रिप्रेजेंट कर सकता है। ठीक है, तो एपिस्टेटिक एलील का काम यही है कि जब भी वो आ जाता है ना, जैसे कि वो दुश्मन है किसी का, तो जब भी ये प्रेजेंट आ जाता है ना कहीं भी कोई भी जेनोटाइप में, तो जेनोटाइप चाहे कुछ भी हो भाई, कोई फर्क नहीं पड़ता, वो जेनोटाइप एक्सप्रेस नहीं कर पाएगा अपना खुद का कलर। ठीक है, सो एपिस्टेटिक एलील आई आई प्रेजेंट था किसके साथ, बी कैपिटल बी डोमिनेंट के साथ, कैपिटल बी स्मॉल बी हेट्रोजाइगस डोमिनेंट के साथ, और स्मॉल बी के साथ। ठीक है, तो आई का कलर क्या है, आई का कलर है व्हाइट, जिसमें कोई पिगमेंट नहीं होते हैं, इसलिए डॉग्स भी बन गए व्हाइट। तो ऐसे ऐसे फेनोटाइप्स उनको मिले थे, ऐसे फेनोटाइप, ऐसे ऐसे फेनोटाइप, और ऐसे ऐसे फेनोटाइप उनको मिले थे। तो जो भी हो, देखिए यहाँ पे ब्लैक बी भी कैपिटल है, तो ब्लैक होना चाहिए था डॉग ना, यहाँ पे भी डोमिनेंट है, ब्लैक होना चाहिए था, यहाँ पे तुम्हारा सॉरी ये ब्राउन कलर को डिनोट करता है, सॉरी गाइस, सो ये ब्राउन कलर को, यहाँ पे ब्राउन होना चाहिए था डॉग, है ना, कुछ तो होना चाहिए था, पर नहीं हुआ, क्यों, क्योंकि यहाँ पे ये वाला जो आई जीन प्रेजेंट था, यहाँ पे जो ये वाला आई जीन प्रेजेंट था। ठीक है, तो यहाँ पे जो आई जीन प्रेजेंट था, यही पूरा काम कर गया। ठीक है, यही पूरा काम कर गया, दैट इज देयर वाज अ प्रेजेंस ऑफ द एपिस्टैटिक एलील which marks the effect of the which repressed the effect of the hypostatic allele. It suppressed, it repressed the effect of the, it inhibited the effect of the allele or the hypostatic allele. देखिए यहाँ पे एपिस्टेटिक एलील में कैपिटल आई और स्मॉल आई प्रेजेंट है, तो सपोज अगर एपिस्टेटिक एलील हेट्रोजाइगस का कंडीशन हुआ तो मैम क्या होगा, तब भी एक्सप्रेस नहीं कर पाएगा, क्योंकि यहाँ पे भी कैपिटल आई वाला जीन प्रेजेंट है, और अल्टीमेटली ये वाला आएगा आपके ब्लैक कलर। ठीक है, यहाँ पे डोमिनेंट वाला था, तो डोमिनेंट वाला पूरी तरीके से होमोजाइगस डोमिनेंट नहीं है, दो आई जीन प्रेजेंट नहीं है, इसलिए थोड़ा बहुत एक्सप्रेस कर पा रहा है ये, इसीलिए ब्लैक कलर का बन जाएगा। ठीक है, तो तब ये कलर अपने आप को एक्सप्रेस कर पाएगा। देखिए क्योंकि यहाँ पे दो आई नहीं है, इसलिए यहाँ पे देखिए स्मॉल आई प्रेजेंट है, मतलब यहाँ पे तो एपिस्टेटिक एलील का कोई काम ही नहीं, यहाँ पे तो एपिस्टेटिक एलील है ही नहीं, क्योंकि यहाँ पे हम डोमिनेंट एपिस्टेसिस के बारे में पढ़ रहे हैं, तो ऑब्वियसली हम हमेशा क्या खोजेंगे, कैपिटल आई को खोजेंगे, तो स्मॉल आई में हमें कोई कैपिटल आई दिखी ही नहीं, तो क्या आ जाएगा, स्मॉल बी जब है तो ये ब्राउन कलर को डिनोट करेगा। ठीक है, स्मॉल आई के टाइम पे अगर कैपिटल बी भी रहता तो भी ये ब्लैक कलर को डिनोट करता। ठीक है, ओके, सो देखिए इतना तक आई होप सभी का क्लियर है अच्छे से। अच्छा, ये वाला भी हेट्रोजाइगस कंडीशन हुआ या कुछ भी, तो डोमिनेंट एपिस्टेसिस में डबल आई ऐसे प्रेजेंट होने चाहिए। आई होप ये पूरी अच्छे से क्लियर हो गई है आप लोगों को। एपिस्टेसिस जो भी मैंने सिक्स कंसेप्ट्स आपको अच्छे से पढ़ा दिए। इंटरजेनिक पहले मैंने क्या पढ़ाया, लेट्स हैव अ क्विक समरी। जीन इज अ सेगमेंट और पार्ट ऑफ द डीएनए. Gene has two main parts, that is known as alleles or allelomers. The gene when interacts with two of the gene, one is known as intergenic gene interaction. Okay, intergenic. One is known as intragenic. Okay, intergenic मतलब क्या, दो genes, when two genes will attract, when there will be interaction between two genes. Okay, between two genes. Intra means when there will be interaction within the alleles of a gene. Okay, within the alleles of a gene. then that will be known as इंट्राजेनिक जीन इंटरेक्शन. ठीक है. उसके बाद इसमें कुछ पॉइंट्स आते हैं. इसमें हम लोग सिर्फ एक ही कैरेक्टर, वी विल टॉक अबाउट वन कैरेक्टर, वी विल टॉक अबाउट वन trait. Okay, here we will talk about many characters or more than one character or more than one trait. ठीक है. यही डिफरेंस है इंटरजेनिक और इंटरजेनिक में. ठीक है. यहाँ पे क्या क्या आती है, यहाँ पे आती है आपकी इंटरजेनिक, मतलब दो दो अलग अलग जीन होंगे. तो यहाँ पे आती है आपकी मल्टीपल अलीलिज्म. ठीक है. यहाँ पे आती है आपके मल्टीपल अलीलिज्म. यहाँ पे आती है आपकी प्लियोट्रोपिज्म. यहाँ पे आती है आपकी पॉलीजेनिक इनहेरिटेंस. ठीक है, पॉलीजेनिक इनहेरिटेंस. इंट्राजेनिक में क्या क्या आएगी आपकी यहाँ पे आएगी कोडोमिनेंस, यहाँ पे आएगी आपकी इनकम्प्लीट डोमिनेंस. ओके, और यहाँ पे आएगी एपिस्टैटिक अली. ओके, यहाँ पे आ जाएगी आपकी एपिस्टैटिक अली. क्लियर. अब चलते हैं आगे. So this was the end of this session. I hope you like this session. If you like this session, then like. पहले तो बिल्कुल लाइक करना, सेशन में बड़ा सा स्माइली बनाना, कमेंट सेक्शन में लाइक करना, शेयर करना बिल्कुल ना भूलना इस वीडियो को अपने फ्रेंड्स के साथ व्हाट्सएप के थ्रू या कोई भी इंस्टाग्राम, व्हाट्सएप, फेसबुक कहीं भी आप शेयर कर सकते हैं वीडियो को. एंड बिल्कुल कमेंट करना आपके डाउट्स अगर कुछ रह गए इस वीडियो में तो उसको कमेंट करके जरूर दिखाना. And thanks for watching this video, guys. Please to support us like this and also subscribe to our channel, that is ANAMIKA'S EDUSPACE. And also don't forget to turn on the bell icon. Please do turn on the bell icon, जो कि घंटी बिल्कुल दबाना. घंटी दबाने से आप लोगों को नोटिफिकेशन जल्दी ही मिलेगी. जैसे मैं पोस्ट करूंगी वैसे आपको नोटिफिकेशन मिल जाएगी. अगर इस वीडियो को अच्छे लगे तो वीडियो को जोर शोरों से शेयर करना, और शेयर करने के बाद आप हमें FB पे और इंस्टा पे दोनों जगह पे आप फॉलो कर सकते हैं हमें. और कहाँ पे फॉलो कर सकते हैं, @the__ अनामिकाज एडुस्पेस लगाना बिल्कुल मत भूलना. तो इसको आप जैसे ही लगाते हैं तो वहीं पे आप मुझे फॉलो कर सकते हैं. और डेली हम लोग वहाँ पे कुछ ना कुछ डेली पोस्ट करते रहते हैं. तो हमारे साथ डेली जुड़ने के लिए FB पर, इंस्टा पे हमें जरूर फॉलो करें. एंड हैप्पी फॉलोइंग गाइज, एंड वी पोस्ट डेली, सो मेक श्योर यू फॉलो अस. एंड बाय ईच एंड एवरीवन. Hope you like this video. Thanks for watching. Goodbye. मिलते हैं. Take care. And next live जल्दी ही आने वाली है, इसलिए चैनल को जल्दी जाके सब्सक्राइब करो. So bye each and everyone. Tata.